All right, good students of AP Bio, I am going to be giving a presentation on DNA replication, which requires a couple of different pieces. So I've split this into 3 videos, hopefully short enough to keep your attention span and get you to take some notes. The first video is just going to be a review about DNA. So we need to have a pretty decent idea of what that stuff is. So let's move forward here to the next slide. And overall, I want to just kind of highlight some of the things on my picture here, and I'll use this model too. I've got my model from school here, so I've got my little DNA model. So ultimately, we want to recall that the DNA is a double helix. So we've got this kind of illustrated over here on this side of the drawing here. That's a double helix. And then if we unwound it, if we untwisted the whole thing and made it look like a ladder, then we'd have this look over here on this side. So like this is kind of, that's that ladder view, okay? Now, the ladder view gives us an idea of how these different components are put together. I just wanted to point out, I'm going to clear that off of there so that we have some room. There we go. This here is a phosphate group. So there's a phosphate group, and it is bonded to a sugar. Now, how can we tell it's a sugar? Because it's shaped like a little hexagon or pentagon, right? So we got a phosphate bonded to a sugar, and then the sugar is bonded to a phosphate. And what's that phosphate bonded to? Well, hey, it's bonded to another sugar here. And then that sugar is bonded to another phosphate. And you get the idea that it goes sugar, phosphate, sugar, phosphate, sugar, phosphate. Over here, they've illustrated that on our drawing over here. They've illustrated that by just pointing out here's phosphate, sugar, phosphate, sugar, phosphate, sugar. So you get the whole idea there that the sides of the DNA ladder are made of alternating phosphates and sugars. And if I grab my little model here and showed you, you'd have your phosphate, followed by your sugar. Trying to get my camera lined up here. There we go. Phosphate, sugar, phosphate, etc. Okay, so the little purple pieces were phosphates and the red pieces were sugars. Okay, the middles, the middle steps, the middle rungs of this thing are made up of 2 chemicals at a time, nucleic acids. And there are 4 types of nucleic acids. So 2 of them at a time. There's 4 total types. Recall that they are abbreviated with either A or T or C or G. So A always pairs with T, adenine always pairs with thymine. So this molecule here will always pair with this molecule. It doesn't matter which side it's on. So thymine could be on the other side like it is here. And then adenine's over here. So A is always pairing with T, and C always pairs with G. That's our pairing rules, okay? So that's the idea of the pairing rules here. Now, those are bonded together, and again, our model does a handy job of showing that there are always 2 colors paired together there in the middle, either the blue with the orange or the yellow with the green. One of them is A, one of them is T, one of them is C, one of them is G. And those make up the steps or the rungs, okay? So that's important to recall. It's also important to note here that the bonds that get built between they're little dotted lines. I'm going to try to draw out some of these or circle some of these here so you can see them. Those dotted lines that exist, those dotted lines imply hydrogen bonding. So hydrogen bonding is a really weak kind of bond. It's not like fully bonded. Electrons aren't being shared. They aren't being stripped away or anything like that. They're just kind of attracted to each other, and they're just kind of holding together just barely. But collectively, when you get a whole bunch of them all working together, then it holds that molecule together. But they're weak bonds, so they're easy to break. That's going to become important, okay? Last thing to note on here is we've noted this 3' business and 5' business. And in DNA replication, this is where it gets really, really important. I'm going to draw out this little hexagon-shaped or pentagon-shaped sugar one more time here. And I'm basically just taking one of these guys, like this guy right here, and I'm putting it, right over here, okay? So we have an oxygen at the top of the molecule, and then we're going to number, remember at every corner there's a carbon, and we're going to number these guys off. There's 1, there's 2, there's 3, there's 4, and then there's a little stub that points up that has a CH2 group on it on the end here, and that's going to be carbon number 5. So there are 5 carbons on this little puppy, and they are numbered 1, 2, 3, 4, and 5. The 5', we name it with a prime. I've never found out why, but we name it with a prime, and we say, okay, that's the 5' end of the sugar, and this is the 3' end of the sugar. And what we find is, if we look over here on our drawing, that this 5' sugar is always pointing up. I'll go up to here. Notice that the 5' sugar is always pointing up. So that's why we label this as the 5' end, because no matter where we break this, this DNA, we break it there, the 5' sugar is going to be pointing up. If we break it here, the 5' sugar is going to be pointing up. It makes no difference where we break it, the 5' on this side is always pointing up. And the 3' is always pointing down. Notice it's always right here, right there, right there. The 3' is always pointing down on this diagram. On the other side, what we find is that it's flipped, it's reversed. The whole thing faces the opposite direction. So the 3' end is facing up on this side, and the 5' knobby is facing down. And so what ends up happening here is we label this side then the 3' end, and this side the 5' end. Is behind my little picture there. Those things matter in DNA replication quite a bit. And so keep in mind what we learned about there was that anti-parallel concept, that the two sides run parallel to each other, but completely opposite directions. Okay, so going to move on to some questions here just for a quick review. Oh, sorry, I got to do this semi-conservative model too. This is a concept that gets thrown at you, and it's a really simple one, but sometimes students learn it the wrong way or are confused on it, and then it's never learned, and it's a super, super simple one. If you have your original segment of DNA, the way that we found that DNA copies is this. The whole DNA thing kind of unwinds and unzips. It basically just breaks apart. It separates. So what ends up happening here is that this, like one side of the whole DNA model, is used as a template for another side to just fill in. So we have this other red side here that's just going to fill in. And what it's going to do is it's going to build new DNA on the opposite side. And remember, if we break it apart and we had, a specific sequence of letters, T-A-A-T, that if it paired up originally with, well, T would pair with A, and A would pair with T, and A would pair with T, and T would pair with A. If you pull it apart and then let new stuff build in, because it's going to follow the same pairing rules, it's going to give you the same thing. So ultimately, one side of the DNA ladder is used as a template. The other side is used as a second template. And then you have new DNA that fills in on both sides. So whenever you make new DNA, one half of it is old DNA and one half of it is new DNA. So when you duplicate DNA, you save half of it. Half, conservative, saved, half saved. So that's the semi-conservative model, and that's how DNA replicates itself. Okay, questions? And then I'm going to pause here for about 5 seconds. So I'll let you pause the video and then go ahead and try to answer these on your own, and then I'll give you the answers in a second. Okay, hopefully you've had a chance to answer those questions. The first one there makes up the sides. That would be alternating sugars and phosphates. The rungs of the DNA structure, those are nucleic acids that make up the rungs, 2 at a time. 4 nucleic acids are A, C, G, and T. And the base pairing rules. If you can't remember, my teacher taught me a long time ago, at the golf course, ATGC, at the golf course. A with T, G with C. That's the pairing rules. A always pairs with T, G with C. And then that 5' and 3' label, hopefully you're recognizing that is identifying which direction the sides of the DNA ladders are going based on their numbered carbon of the sugar. So think about what that means. It's the direction that the DNA ladder's sides are going based off the numbered carbon that they're facing. So that's what that means. All right, that's part 1. Move on to part 2 when you're ready.